Hey everybody, we're going to have a look now at the cheery topic of rigor mortis. Before we do, we have to talk about some aspects of myosin and actin interacting, and then really throw ATP back into the conversation. We discussed this somewhat in class, but let's make sure we really understand what ATP's role is in muscle contraction, because that relates to what happens in rigor mortis. So, let me get out of the way here. Let's review the cycle of the forming cross bridges, contracting and relaxing, so you understand the role of ATP in particular. Let's start up here with this diagram. What we have here is we have myosin is in this high energy configuration. We've used some energy. ATP has been hydrolyzed rather, and we'll come back to that meaning in a second. But ATP has been used so that myosin can be in this position to grab actin. Remember these little black dots we see here? I kind of messed with my arrow. These black dots right here are the myosin binding sites on actin. The blue chain you see here, that is actin. Also recall we have the yellow pieces and then these tan spaghetti pieces. This tan spaghetti, remember, this is tropomyosin. These are normally blocking the binding sites on actin. They are normally in the way. However, to have a contraction occur as we're having here, calcium must be available. Calcium bound to troponin, these yellow protein complexes, which then roll the tropomyosin out of the way, so myosin can now grab it. So we start with this premise. Calcium is available. We're signaling a contraction. Myosin is in the right position. It has oriented because energy was spent to do that. It's now bound to the actin. So what happens because of this, we're having A, D, P, and P. Remember, these represent energy molecules. Phosphate, let me grab my pencil again here. The phosphate is going to be released. When that releases, that actually is basically giving away some energy, and that causes that change in the position of myosin, so that's what we associate as the power stroke. Remember, it's bound to actin, so when it changes shape, it pulls on actin, and then at the same time, as a result of this, we're going to release ADP as well. So now we have everything in this position. Note that myosin is still bound to actin, but now we've moved actin a little bit. We've had this power stroke. But now we come back to this. We have to have ATP available to cause the release of myosin from actin and to reconfigure it. So let me go back to my verbiage here. So ATP comes in, myosin releases actin with the help of ATP and enzymes. Essentially, we're investing some energy. It causes the break in that to occur, and that allows myosin to break free of actin. So ATP had to be there for that to occur. Enzymes also help the process. So ATP will then be split. That is the hydrolysis process. When you lyse, you break. So we split ATP to ADP and P, and then that allows us to reconfigure to put back into position myosin. Remember, it was laying on its side. Now we're going to put it back in position so it can grab actin again. So once we have established that, the ATP was used to position the myosin head to make sure it's in the right place. When the phosphate will then be released, as we just did a little bit ago, it will change shape and cause it to pull. And as long as calcium is available, and as long as ATP is available, we can keep the cycle going of changing shape, causing the pulling action to occur. Then ATP comes in, we release, reconfigure, change shape, release, reconfigure, change shape, etc. So this is the action we associate with a muscle contraction. Note, you have to have calcium, you have to have ATP, and what's implied here, but you don't see it on the screen, is you had to have oxygen. Oxygen is used to make ATP. Remember, we use glucose and we use oxygen in our mitochondria to make ATP. So if you have calcium and you have ATP, and you have to have oxygen to have ATP, you get to have this cycle as long as the signal is being sent to the muscle to contract. Which brings us then to rigor mortis. Rigor mortis, and you see down here what rigor mortis really looks like, are these contractions post-mortem. So after someone has died, there can be some contractions of their muscles. And that sounds really odd, but it can happen for very logical physiologic reasons. The muscles are going to begin to stiffen. I have 3 to 4 hours. It can be as little as 2 hours after death. And rigor is most pronounced. That is, the muscles are stiffest after about 12 hours, and this can last 48 hours, even up to 60 hours. So someone dies, and then a few hours later, their muscles contract. And that sounds strange, but why does it happen? Remember the ingredients for a muscle contraction to occur: calcium, ATP, and oxygen. Well, what's going to happen when someone dies is those dying cells of the sarcoplasmic reticulum and of the cell membrane of the sarcolemma are going to begin to break down, and calcium begins to enter to the muscle fiber. Calcium leaks out of the sarcoplasmic reticulum, or it leaks in from outside of the muscle fiber, and calcium is now available. Tropomyosin rolls out of the way, and myosin, which is in position, is going to grab actin. And there might be some available ATP to even facilitate this. So this activates the myosin-actin cross bridges, muscle contracts, but it can't release. If there's no ATP available, and why is there any ATP available? Because if you are dead, you're no longer breathing, and therefore you can no longer make ATP. Myosin is going to bind to actin, but it can't let go. So someone's going to experience rigor mortis. The muscles are going to stiffen, and they're going to stay stiff until actin and myosin, the myofibrils, begin to break down. So that's why it happens after death, that calcium leaks out of sarcoplasmic reticulum, or leaks in from outside of the muscle fiber. Calcium is now available. Tropomyosin's out of the way. Myosin can bind actin. It can even kind of pull in tension a little bit, but it can't release without ATP being there. So it's going to be most pronounced after 12 hours. People typically are going to have the smallest muscles go through rigor first. The larger muscles may go through rigor later. Even internal organs are going to have some rigor. That is, there will be some contraction of some muscle fibers. And this can last up, again, 48 hours, even 60 hours. This is important forensically because it helps us establish time of death. If someone discovers a body that is warm but is not stiff, that is, the body still has its temperature but hasn't gone into rigor, we can tell even from that they've probably died in the last hour or two. However, if they're warm and yet stiff, they've gone into rigor. So now the time of death is going to be later. It could be probably 5, 6, 7, 8 hours. As the body cools off, obviously we can start to look at a different time interval. Someone can be then cold and stiff. That is, they might have died the day before. So they're cold, they've lost their temperature, but they're still in rigor. And then if they're cold and not stiff, they have come in and out of rigor already. So now we know they have been dead for 2 or 3 days. So this is one of the ways that they get an estimate for someone dying, to try to establish what's called the post-mortem interval, the PMI. Please do not believe that on CSI and all these crime shows they can accurately predict this. There really isn't a good single factor that helps us determine exactly when someone died. If someone dies 5.5 hours ago, they will be warm and they will be in rigor, but we can all do is establish an estimate. We can't say they died 5 hours and 27 minutes ago. There have been lots of investigations looking at the vitreous humor of the eye, looking at liver temperature, looking at all kinds of variables. And we can get some fairly good estimates, but nothing as precise as you hear on TV. Why do we mention this stuff in A&P? Rigor is an important process to understand, because if you are working in a hospital and there is a patient who dies and is in rigor, and the family were to see the stiffening or to see the person's finger move, they may get a little freaked out because they thought the person had died. These contractions are not uncommon for someone who passes to have these contractions occurring again from 2 to 4 hours after they die, up until to be stiff 48 or 60 hours afterwards. So please understand that this is not a strange process. This is a process that's born out of changes in what was really a function of muscle contraction. The fact that calcium is available and ATP isn't allows contractions to occur but not stop. Thank you very much. That's it.